this one open. Let's go to chapter 1. Let me just skim through this really quickly. Oxidation. Omega. Okay. One of those. Yep. I know some of these words. Skim reading really quick. Okie dokie, y'all. Question number 1. There, this here. Good. All of our glycerol-containing phospholipids are synthesized from what? Cardiolipin, phosphatidic acid, mevalonate, or ceramide?
Isn't it B?
The answer is B. Phosphatidic acid. That's going to be one question. And this answer pops up a whole bunch a lot. So phosphatidic acid is literally the smallest and the simplest phospholipid. So, and you'll see this a lot. We talk about whenever we talk about fats and lipid synthesis. You can call them lipids or you can call them fatty acids. Okay, so you're going to see that a lot. Kind of so fatty acids or lipids, kind of interchangeable. You'll also see kind of words like fatty acid chains, that kind of stuff. Or they'll call them free fatty acids. In Biochem 2, especially at the beginning, there's a lot of like AKA type words where there's a lot of different words that all are saying or mean the exact same thing. You just kind of have to get through all of them and remember and learn the vocab portion of it. And let me actually see real quick if I can make sure that I don't miss anything. Cardiolipin. Pretty sure it mentions that in the book. It doesn't go super deep into it. Cardiolipin is a phospholipid, which is exclusively in the mitochondria. Which he does get into mitochondria stuff. Mevalonate. Mevalonate is also known as mevalonic acid. And we'll talk, you'll get to, I don't know if have you guys gotten to cholesterol yet?
Yeah.
Yes.
Okay. Have you seen mevalonate? I know that that's going to be within the cholesterol lesson. You'll see that. Was that last week or is that this week?
It's this week.
This week? Okay. If you haven't done that stuff yet, you should see mevalonate. There's a mevalonate pathway that you'll get to with all of that stuff. And then the ceramides. Ceramide is what's known as a sphingolipid. Have you guys heard that word yet?
Yes.
Okay. Ceramide doesn't come back a ton, and sphingolipids, honestly, like I barely remember even hearing the word sphingolipid. I remember that I think there was one question at some point about sphingolipids, but not like crazy important subject for right now. And I think this is the same question back to back. Yeah, it is. So go away. Next question. Biosynthesis of palmitic acid does not use which of the following? Biotin, bicarbonate, NAD+, or acetyl-CoA.
Pretty sure that's NAD plus.
Yeah, NAD+ is the correct answer. It does not use the keyword not. Palmitic acid. Make sure I grab all the good stuff. The reason why palmitic acid is important, and you'll see a lot of palmitic acid. So two really, really big ones, a lot of like test questions that come from this chunk of material. He's going to hit phosphatidic acid a lot, and he's also going to hit palmitic acid a lot because he really, really likes those two. And because again, it's one of the palmitic acid is the most common saturated fatty acid that's found in the human body. So it comes from a lot of animal type stuff. And this is also, if you think about this, if we know that palmitic acid is from animals, biotin, let me remember what biotin is.
B5.
Yeah. The active, actually, it's B7. Biotin is B7. It's one of our B vitamins, right? Biotin is B7 because 5 is, what's its tall, rich, nudists, played pickleball for centuries. That is 1, 2, 3, 5, 6, 7. I'm missing one. Tall, rich, nudists, play pickleball for centuries. Jeez. Tall, rich, nudists played pickleball for centuries. There it is. Biotin B7. 1, 2, 3, 5, 6. That's where I messed up. No, 5, 6, 7. Yeah. What am I missing? Tall, rich, nudists played pickle. That's why. Pickle ball is 7, 9, and 12. There. You don't remember this little mnemonic? Really, really, really important for boards to remember the B vitamins. You want to remember that B vitamins come back a ton, especially when you start talking about like pathology stuff. B vitamins come back over and over and over again. Bicarbonate has to do with our CO2 stuff. And again, when you talk about breaking down into organic matter, breaking down fatty acids, bicarbonate is going to be one of the things that comes off. And then acetyl-CoA, we're going to talk a lot about acetyl-CoA. You'll more in like the fatty acid beta oxidation breakdown of fatty acids kind of stuff, which I'm sure y'all have already seen since we're a little bit behind. But any questions about palmitic acid in general? Nope, moving along. By what mechanism is palmitoyl-CoA, or by what mechanism does palmitoyl-CoA regulate fatty acid biosynthesis? I'm going to pull up my slides because, like I said, it's been a little while since I've looked at the beautiful biochemistry. Any thoughts on this one? So we have It regulates fatty acid biosynthesis by negative feedback regulation. It is converted to stearic acid, and stearic acid transcriptionally regulates fatty acid biosynthesis. It activates the polymerization of the acetyl-CoA carboxylase and thereby increases palmitic acid biosynthesis by phosphorylating the rate regulatory enzyme in the fatty acid biosynthetic pathway. There's a lot of big words there.
I'm guessing A.
Any other guesses? A is correct. It regulates the fatty acid biosynthesis by negative feedback regulation. I want to find that in the slides. Here is good picture. Uses one in his book. If I can find it. There it is. Our palmitoyl-CoA regulates. This is our one of our very first steps. And if we don't have palmitoyl-CoA, eventually, finally, when we go all the way through and get our acetyl-CoA, we're talking about synthesis, other side of things. I can't find it in the book. That's okay. We'll just keep going. CDP diglyceride is not involved in the biosynthesis of which of the following? Phosphatidylcholine, phosphatidylserine, phosphatidylphenolamine, or sphingomyelin.
D.
That is correct. It is sphingomyelin. CDP diglyceride. It's an intermediate which occupies a branch point in the lipid metabolism. Let me see if I can find. Man, I had so many good resources before at one point with all this stuff. I don't remember where they all ended up. But you can see our CDP, the CDP diglyceride. Kind of, you can see in a lot of this stuff, it acts as kind of that branching spot where it really can go onto a whole bunch of different sides of things. In this case, we look at the sphingomyelin. what do you guys know? What do you guys talked about with sphingomyelin? I feel like these questions aren't super based in the first chapter of the book. I was looking back over it earlier. I don't remember seeing any of this.
I don't think we went over sphingomyelin.
Yeah.
Yeah, I don't think we went over it too much. He glanced over it,
I'm like,
but I haven't looked over week 1 yet, so.
yeah, week 1 of the book is not as like a lot of this stuff isn't in week like first chunk of the book. I'm a little bit confused why the Linus questions are jumped ahead so much. Let's do this instead, because I want to spend a little bit of time. I think it's gonna be more beneficial. How do you guys feel overall about the material? Let's start there.
There's so much.
Not
It is a
the
lot.
greatest.
I would say don't feel real good about it.
Okay. Let's do this. I know I already kind of do this in the videos. But let's just start in the book because I don't like the Linus learning questions right now because I feel like they're jumping all over the place and they're further in information than necessarily what is talked about in the first chunk of material. So let's just start, going through. Let's start by going through a little bit of the reading material, looking at everything. As we go through this, I'm gonna skip around a little bit. I'm gonna try and just go straight through. Might skip around a little bit. As we go through this, though, hit me with questions. Like if something doesn't make sense or something is like, the heck is going on there? Let's stop and talk about it because, like I said, Biochem 2 becomes a big back burner course, and I think that doing this is going to be much more beneficial for us. Okay, first things, learning topics. Digesting of fats, how the fatty acids are mobilized and then transported in tissues for oxidative degradation, so breaking them down, how fatty acids are oxidized, and then ketone bodies. Yeah, in this whole chapter, there's nothing that's almost none of the questions that we've talked about have been in this first chapter. So I don't know if Linus learning is maybe it's out of order a little bit or what's going on, but
If those are the ones I sent you, I may have labeled it wrong. I'm going through them right now to see. I may have just saved it as the wrong chapter.
These are the ones that Kristen sent me because I just re-downloaded them.
Okay.
So I don't think that they're wrong.
Like, because going through my Linus ones just from chapter 1, like just on the app, it's completely different questions.
Yeah, I don't know what went down. We'll figure that out in a little bit. Okay. So talking about fats in general, a little bit overview of fats. Fats are incredibly important. If anybody's ever told you that you need to eat less fat in your body, they're lying. Fats are incredibly important for a lot of the things that we need to do. They're very important for a lot of our hormones, a lot of balance, a lot of our immune system is based off of fats. So, and additionally, they are an incredibly important energy source. Granted, too much of it, too much of a good thing is always bad. But fats are very, very important. They're important in general health and nutrition. They provide the thermal insulation to our tissues and organs. Lipoproteins serve as a cellular constituent in membranes. So a lot of our membranes, the phospholipid bilayers, all of our membranes are made up of fat, including the cytoplasm, the mitochondria, and then also lipid transport. Okay. They're required for the steroid hormones. Remember some of those main steroid hormones that we talked about, the testosterone, some of the thyroid hormones, estrogen, progesterone, all of those guys. Remember these guys, this sometimes comes back. Remember that those are our nuclear, they bind to nuclear receptors inside of the nucleus. So lipid or fat metabolism includes both their breakdown, the catabolism, or the biosynthesis, anabolism of the fats. Many of these lipid molecules are also important signaling molecules. All of our stuff that we talked about was signaling, and they play the major roles in cell signal transduction. The cell signal transduction, a lot of that is going to be seen when you get to the chapter of eicosanoids. I don't think you guys have got there yet.
No, not yet.
Yeah, that's either, let me check. It's either beginning of exam 2. Yeah, that's the very beginning of exam 2. Okay. You'll see I'll talk a lot about eicosanoids at the beginning of exam 2. Certain defective metabolism of lipids can produce some diseases. For example, atherosclerosis. Have you heard of atherosclerosis before? Do you know what it is?
Yeah, isn't that like hardening of the arteries? Calcification or something? Mm-hmm.
Yeah, essentially, next trimester, you guys will be in Gen Path, and you'll have a whole big chunk on this, and this will come back in a couple different classes. I'm going to make this super, super simple for you guys. Not needed for this course, but later on. Atherosclerosis, essentially what happens is you have your arteries, and along the walls of the arteries, fat comes and essentially embeds itself along the wall of the artery.
Mm-hmm.
And it can actually invade down, because you remember, we think back to kind of the whole structure of the artery, you have like the tunica externa, the tunica media, and the different muscle layers and all that kind of stuff, the elastic layer of the artery. So you actually get some of this fat can sneak inside of that innermost layer and start to build up, and it builds and it builds and it builds and it builds. When it gets to this point, specifically if it's in the heart, that's coronary artery disease, because you get buildup and it can block blood flow.
Mm-hmm.
But the biggest issue with this is what can happen is our innermost layer of our artery can actually rupture and break because of this buildup of fat. And when that ruptures and breaks and pops up, all of a sudden, all the blood that's flowing in through here can sneak into this side here, and it starts to form a clot. And then you can actually get a clot of the vessel. And then that's called a thrombus. If a thrombus breaks off, it's called an embolism. Pulmonary embolism, you can have an embolism in the brain, all that kind of stuff. That's what happens from atherosclerosis. Again, not for this course, but later on. Gen Path next try. What's it called? The physical diagnosis. You'll talk a lot about thrombuses and arteries and heart stuff. So this will come back a lot.
Okay.
So atherosclerosis, obesity, digestive disorders, to several other complicated cardiovascular dysfunctions. A lot of gallbladder issues. You ever meet somebody who has had their gallbladder taken out?
Mm-hmm.
They're super, super strict about their diet. They can't eat any fat, otherwise they have to run to the bathroom. So that's because the gallbladder is what releases a lot of the chemicals Our lipases, which help break down fats, are made and stored in the gallbladder.
Mhm.
Okay, so starvation and diabetes mellitus. I'm going to take a second really quick here. Again, a little tangent. Do you guys know what the difference between diabetes mellitus and diabetes insipidus is? Have you talked about that at all?
No, but diabetes mellitus being a problem processing sugar in the bloodstream, whereas diabetes insipidus, I'm assuming that I feel like I've heard it before related to kind of like frequent urination or something.
Yeah, diabetes insipidus, something like that. I'm missing an I. That has to do with, you're exactly right, with water, the way that our body stores water. It's a pituitary gland issue. The gland that sits in your skull, kind of like right here inside of the sphenoid bone, inside of the sella turcica. It produces anti-diuretic hormone, or ADH. And when you have a whole bunch of issues with ADH, that's when you get diabetes insipidus. That's going to be super big in next trimester in Phys 2. You spend a ton of time talking about diabetes mellitus, ton of time talking about diabetes insipidus. A lot of people get them mixed up, get them confused with each other, and it's a lot of questions on exams. So that's coming up in the future.
Okay.
Back to fats. Starvation and diabetes mellitus. So with starvation or with diabetes, because with diabetes we know the issue is that we can't get the sugar into the cells because it's an insulin issue, right? We have an issue with the insulin, whether we're not producing any insulin, like type 1 diabetes, or because our cells are super desensitized to insulin, as in type 2 diabetes. So our cells, specifically our brain, is not getting the glucose into the cell that it absolutely needs to have in there. So what happens is we increase fatty acid oxidation. Oxidation in this case means breakdown. So we're going to be breaking down the fatty acids, the fatty acid chains, and we produce these guys called ketone bodies. Ketone bodies, if you've ever heard of the trend diet keto, that's what it's short for, where you don't eat, or the Atkins diet is another version of it, where you don't eat any carbs and force yourself into ketoacidosis. Carnivore diet is another one of these that essentially does the same thing. A lot of these can be good if like done correctly and supervised under doctor supervision for short periods of time. The body's not meant to be in ketosis for long periods of time. And when we're in ketosis, after we're in ketosis for a long period of time, we call it ketoacidosis. A question that comes up, a test question that comes up that he likes to ask, I think this class it'll get asked, and then also in future courses, they like to ask it this way as well. They'll describe a fruity smell on the breath.
Okay.
Where, and literally, if you go up to somebody, like if somebody has, if somebody does have like diabetes or something like that, and you can go up to them, they pass out or whatever, and you smell their breath, and it smells super, super fruity. It smells sweet. That's a sign that they're in ketoacidosis, and they need the help right away. So,
or it's gonna smell like acetone.
Yeah. Have any of you ever watched The Amazing Dr. Pol, the veterinarian show?
No. Oh, yeah.
Yeah, he talks about this a lot. Cows have a lot of issues with ketoacidosis. A lot of times, it's one of the big like reason why a vet goes out to help them with that, get them like the proper nutrition and stuff that they need, or their get sugar IV and stuff into their blood.
Oh, interesting. Yeah.
Yeah. And so all of these things that are happening, and whenever we're in starvation, whenever we have ketosis, whenever ketoacidosis, it's going to promote gluconeogenesis. And if you remember, gluconeogenesis is that production, so making glucose out of non-carb sources. So using our fats, using our proteins to convert them into glucose. Any impairment or dysfunction in the fatty acid oxidation leads to hypoglycemia. Please, please, please make sure you pay attention to these. Anytime you see a word that's hypo or hyper, make sure you write that down somewhere or highlight it or something, because all of those words, hypoglycemia, hyperglycemia, all that kind of stuff, big test type questions. Not just in this course, but in a lot of courses and like pathology courses and stuff. They love to ask questions about hypo this, hyper that. So really, really pay attention to this. In this case, hypoglycemia, meaning low or not enough blood sugar. And also deficiencies of carnitine or enzymes can cause defective fat metabolism. We're going to talk more about carnitine in a little bit, but essentially, carnitine is the protein that really, really helps us, the body, actually turn that fat into energy. We'll talk about that in a little bit. The fat supplies competent fuel stores. So here's where we're going to start seeing some words. Triacylglycerols. A lot of people get really confused with this stuff because they see words like triacylglycerol, triglycerides, fatty acids, or they call them glycerol fatty acids. If you see any of these these words, is it spelled wrong? Triacylglycerol. If you see any of these words, they're all talking about the exact same thing. They're essentially, there's like, I think like from definition wise, there's like little variances, but for the purpose of this course, these all mean the exact same thing. Don't freak out. Like triacylglycerol versus triglyceride, like they mean the same thing. They're just different ways of saying the same thing. So oxidation of fatty acids is a major energy yielding. Remember, there's 9 calories per gram of fat. About 1/3 of our energy requirements are obtained from dietary triacylglycerols. 80% of energy requirements of mammalian heart and liver are met by oxidation of fatty acids. Many high-rating animals depend almost totally on fats as their source of energy. Here is our root of our fat. So you're going to see this, and you're going to want to get used. There's a whole chunk. I don't remember which lesson it is. You might have already talked about it. It might be in the lipid synthesis, where he has a bunch of pictures of different fatty acid chains. And he's just like, hey, know all of these. Let me see which one it is. I don't know if it's here or if it's later on. Might be in the book. There is a whole chunk with just fatty acids that honestly it's just straight memorization. We'll get there when we get there. The basic structure is right here. The fatty acids, the way that they look, they go in a chain, in a carbon chain. So you have a carbon like this stacked. N right here represents the number of carbon atoms. So like we looked earlier at the palmitic acid. That one was a 16 chain, so there's 16 carbons. So we look and we here, and then off of every single one of these, you might see it drawn like this. If you ever see a line that represents a bond that doesn't have anything on the end of it, that has hydrogen on it. And then at the very end, we finally get It was C. Boop. Like that. And then we have an R group off on this side. A lot of times being, when you bring them all together, it becomes that glycerol. And it becomes a triglyceride. So general structure of fats, you'll see this a lot. There's a component where he'll just have you, like I said, you just have to name a whole bunch of the different fats based on like the number that they have, or kind of like the rough structure. So that'll come up. Y'all's test for this course is it 2 weeks or next week?
I think it's 2 weeks.
Okay, yeah, I think that fits.
So we got a little time.
Yeah.
Thank goodness.
Yeah.
We'll try and get through a lot of this.
Yeah, next week it's gross anatomy, phys, and micro.
Nice.
Yep.
Okay. Continuing, so fats have advantage over polysaccharides. Remember, polysaccharides, those fatty acid, the glucose chains, multiple glucose stuff, multiple sugars, multiple carbs. Fatty acids carry more energy per carbon because they are more reduced. This is a point that comes up a lot when something is more reduced or more oxidized. Okay, so just more reduced, meaning that it has more hydrogens. So because it has a lot more bonds, remember we talked way back in Biochem 1, a bond, literally all it is stored chemical energy. So because we have a whole bunch of bonds and stuff that we can break through here, we have a whole bunch of stored energy. Fatty acids contain less water alone because they are non-polar. Glucose and glycogen deliver the quick energy. They provide the short term, but fats provide for the long term energy needs. Can be stored for a longer period of time and provide slow delivery of the energy when needed. The dietary fat are absorbed in the vertebrate small intestine and then are packaged and transported by a typical lipoprotein called chylomicron. This is going to be a test question a million times over. It always asks about chylomicron, okay? So the question will literally be almost this exact kind of phrasing, like what's responsible for packaging and transporting lipoproteins or lipids or whatever. Chylomicron. I think there's a cool picture.
And aren't these usually transported via the lymph instead of like the
Yeah.
hepatic portal vein and stuff?
Yeah, they will be. So what happens is, actually a lot of our nutrients and stuff are transported by the lymph, but especially fatty acids. So you have this cool little. This is what a chylomicron looks like. It's almost like it's just like this big ball. And so what happens is in our stomach we have the different enzymes, or specifically in our intestines. When we go from the stomach, it gets broken down by some acid, injected into the small intestine through the pyloric sphincter, and the lipases and the proteases and all that kind of stuff from the pancreas and from the gallbladder mix, and they start breaking stuff down. And then you have a whole bunch of, I think you guys should have talked about this in BCT, where the walls of the small intestine have all these microvilli, right? And actually, technically, if you look at it, there's actually like even smaller stuff like this.
Wow.
And so what actually happens, so all of these broken down little molecular bits and pieces get passed through and sucked up in through here. So chylomicron is this big ball. It's going to be waiting right in here. And it's going to grab those fats and tuck it inside of it. And then, yeah, it'll travel through the lymph. And the lymph has a couple different points where it eventually dumps into typically the venous side. And so when it dumps back into the bloodstream, that's when then, okay, it has all the stuff now in it, and eventually going to travel to the liver. The liver is really, really responsible for a lot of the storage of energy. There's a lot of our glucose stored in there and stuff, as well as it'll help store a lot of fat and help process a lot of this kind of stuff. Okay, so we have chylomicrons from the small intestines into the blood, eventually goes to the liver, and then it gets stored in these lipoproteins. We call them very low density lipoproteins, or VLDLs. Have you guys talked about, it's probably this week, the VLDL, LDL, HDL?
Not
A
so
little bit.
much. Okay, maybe I
I think
missed it.
that's this week. It's in the cholesterol
We did
stuff.
in our one-on-one today.
Okay, so you talk about this stuff kind of this week with cholesterol. So the chylomicron, once it brings it all the way to the liver, it drops it off in the liver. And there's these other lipid lipoproteins. So it's essentially just a protein with lipids mixed into it. And they start off, like, they're pretty similar. Let me find a good picture of VLDLs. See, so they look very, very similar. VLDL, IDL, these guys. Okay. So VLDL is, again, it's another lipoprotein. The chylomicron will pass off all of the fat to the liver where you have these VLDLs. And essentially what happens is the VLDL turns into, I don't remember if he mentions the IDL or not. I think he does. So it goes from VLDL to IDL to LDL, and we just group these together as HDL. Those being the very low density lipoproteins, low density lipoproteins, and high density lipoproteins. Essentially, the more fat and stuff that we pump into that protein, that lipoprotein, the denser it gets and the more fat that there is, that it's holding, that it's containing. So we'll get to this eventually later in the future, but these very low density lipoproteins, because fat is such a great energy source, we need to get it to where it needs to go. What happens is you take all of this stuff and you pack everything into the VLDLs. And part of the job of the VLDLs and all of these LDLs, high HDLs, is to go out into the blood, and it's going to go around to all the different cells and drop off lipids and fat wherever it needs to go. And that's why when you do blood work, eventually when you get to your lab diagnosis course in Tri 4, you'll look at the actual values of this and kind of what's good. Easy way to remember: VLDL, good. We want high VLDL. High LDL is still good. These are both our good kind of cholesterol. And then, but if we have high HDL, that's bad. HDL is what's left essentially after when the lipoproteins circulate through the blood, dropping off lipids. If it doesn't have a lot of lipids to drop off, like if there's excess fat, you get more high LDLs, high HDLs. You get more HDLs present in the blood because it has a bunch of extra fat. And that's the way that we actually measure our cholesterol, by looking at this. So if you have high LDL, that's good. High HDL, that's bad. That means you have too much fat within your blood, and you're at risk for those other diseases and stuff that we talked about. Because when there's a lot more fat in the bloodstream, you're more at risk for that atherosclerosis, the obesity, all that kind of stuff. Okay. And that'll be when we get to the cholesterol stuff. So fatty acids are esterified. Who remembers what that word means?
So an ester bond, is that like it's bonded to an oxygen singly in one part, and then for the rest of it, there's another. Is there like another, like a keto group attached to, or no?
Something kind of like that. The big thing with ester bonds is they use OHs. They use alcohols, the hydroxyl groups. The ester bonds use the hydroxyl groups, and we'll talk a lot about the ester bonds with fatty acids. So keep that in mind.
Okay.
Whenever we're talking about fatty acids, 9 times out of 10, if it asks about a type of bond, it's probably going to be an ester bond. So an ester bond forms between the glycerol to produce the triacyl glycerols. So right here is our fatty acids. Sometimes these will also be called, before they're bound, they'll be called free fatty acids, or they'll be lazy and write them FFAs. It all means the same thing. Free fatty acids, FFAs, all saying the same thing. And then when we make them, we have triacylglycerol. What's another name for triacylglycerol that I told you guys?
Triglycerides.
Triglycerides. Yeah. So don't get confused about that. Professors love to interchange all of these, and biochem, a lot of his test questions interchanges them. So just remember that they're the same thing. So we have esterified. Esterification gives us, as part of the synthesis process of triglycerides, hydrolysis is going to give us the fatty acids and the glycerol, which then we'll use for beta oxidation when we need to break stuff down, which we'll talk about later. Lipoprotein lipase, a hormone-sensitive enzyme, catalyzes the reaction and cleaves triglycerides or triacylglycerols to release the free fatty acids and glycerol. Again, this is the beta oxidation type stuff that we'll get to later.
So earlier on, there was a section that talked about phospholipase A1, A2, phospholipase C and D. Is that what he's referencing there? By your estimations?
Yeah, the lipoprotein lipase. Yeah, that's going to be these guys.
Okay.
The phospholipases are going to be our lipoprotein lipases.
All right, just making sure. Thanks.
Yeah. And we're about to get to those as well. Catecholamines, please remember this word. Catecholamine are dopamine, norepinephrine, epinephrine, these guys. These come into play a lot, not just in this class, but a lot in the future. The importance of dopamine, norepinephrine, epinephrine, all of this stuff comes back constantly. You'll see it a ton in Phys 2 and pathology courses from here on out. So just remember catecholamines and remember these words. Okay, so catecholamines. You have a question?
I had a little minorly tangential callback to these catecholamines and their action, because I was reading through this chapter and the next one and stuff, and they're talking about the activation of protein kinases. So is this they're referencing the G protein coupled receptor system, and isn't this the case where that then transitions to cyclical AMP, the CAMP, and that is what activates the protein kinase to then trigger activation of the lipolipid proteins? That wasn't right. Lipoprotein lipases.
Yeah. So remember the whole G protein coupled receptor stuff.
Yeah.
All of. Let me find a good picture of it.
I mean, I see that he referenced it there, but I was just like, oh man, okay.
And
see if I can remember.
That comes back. You're gonna get test questions about the G protein coupled receptor.
Yeah.
And how that activates, and you have the 3 different components of the G protein, the alpha, beta, and the gamma. Yeah. And how the beta and the gamma come off of it so that the alpha component is activated, and then that activates the cyclic AMP, all that kind of stuff. And you get all of this. Then it's this comes back. If you remember last try when I was doing tutoring, if you were in those sessions, I mentioned that. I told you guys it would come back.
You sure did.
Too,
and here we are.
And here we are.
Yeah.
Every now and then I know what I'm talking about.
Yeah. At least twice a day, like a clock.
Exactly. A broken clock. Even a blind squirrel eventually finds a nut.
Yeah, exactly.
Okay, so catecholamines and glucagon are able to activate the hormone-sensitive lipase through a series of reactions. The catecholamines mediate their actions via binding to their respective guanine nucleotide binding protein, or G protein. Occasionally he refers to it as guanine nucleotide binding protein. Just remember G protein is guanine, and you should be good to remember that.
All right.
So the G protein coupled receptor systems, followed by activation of the protein kinase, and then phosphorylation of the hormone-sensitive lipase, and so on, just like you were mentioning. Yeah, so the catecholamines activate the G protein. The G protein phosphorylates the lipoprotein lipase or the different phospholipases, and that's what's going to actually start to cleave the triacylglycerol by different phospholipases. So he will have test questions. It's like this picture right here. And I'm sure we'll see them in Linus Learning. I'll figure out the Linus Learning thing, why the questions are out of order or whatever's going on. You will see that he'll ask specific questions about these different phospholipases. So you'll get a question specifically that is like, which of the following phospholipases breaks the bond of the oxygen and the carbon on the first chain, being A1. He loves to ask about phospholipase C. The ones I've seen most common are A1 and C that he's asked about. For sure, C likes A1, occasionally A2, but yeah, most of the time phospholipase C. But you'll want to remember the position of each of these, where it is that it's breaking off.
Okay.
Let me see real quick. Okay. I was thinking that we were going to jump into more of the synthesis side of things, but I guess we can do the oxidation first. Okay, so phospholipases. So we formed it, now we're doing the hydrolysis, we're breaking everything down. All of these guys, they hydrolyze all these different points. Glycerol is going to enter glycolysis to produce pyruvate. Remember, if we think way back to our glycolysis, remember the
that cycle in the liver where it was like formation of fructose 1-phosphate or something like that, and then it had a side
kind of like, yeah, glycerol goes in, it enters in that the DHA, you shorten it as the DHA. DHA, the dihydroxyacetone phosphate, kind of where in glycerol, it normally, when you get the cleavage of the 6 carbon into 2 3 carbons, and you produce the glycerol 3-phosphate and the dihydroxyacetone phosphate, and then they both get converted. Sorry, you do the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and then the dihydroxyacetone phosphate goes through another couple steps to turn into glyceraldehyde 3-phosphate again as well. Kind of that whole process where you go from 2, 3, 4, 5, 6, and then it splits into the 2, 3 carbons, the DHA and the glyceraldehyde 3-phosphate, and then this turns back into glyceraldehyde 3-phosphate, and then you continue down to get 2-pyruvate. So that's kind of where glycerol gets chunked into here. Again, you're going to want to, this is another one where you're going to want to make sure before test time, before next week, write out the steps of this, because you're going to have test questions about enzymes, about kind of where this fits into it, what turns into what. You'll have a test question or two about that little process right there. And here's just the written out step-by-step of that. So kind of the whole point of all of this, the free fatty acids. So this is where the glycerol goes. The glycerol gets broken down into glycolysis to eventually form pyruvate. But as well, the free fatty acids are going to be catabolized. Catabolism means to break down to yield acetyl-CoA. They're transported in the blood has the FFAs. Plasma albumin is the major carrier of the free fatty acids in blood. That's another one I want to remember. That's where he'll get you with questions where you'll have two different questions. He says, what is the carrier of free fatty acids in the lymph? And that is going to be, what's the name of that protein? The lipoprotein that does it in the lymph?
Albumin.
Oh, that's chylomicron.
Yeah, the chylomicron.
Oh shit.
Yeah, that's why you remember, got to make sure pay super attention because he'll have that. He'll have lymph and he'll have blood. So like, what's the difference between the, you have the chylomicrons and then you have the plasma albumin. Yeah, and that's for the free fatty acids. So the enzymes that degrade the fatty acids through an oxidative process are localized within the mitochondrial matrix. What else occurs in the mitochondrial matrix that's super important about energy production?
The electron transport chain?
That's within that inner membrane.
In the membrane?
We also have that Krebs cycle on the inside. So yeah, both of those guys. So this makes sense. If we think about it, the whole point of fatty acid and like breaking it down is to give us energy.
Mm-hmm.
So our end goal with this is to create a bunch of acetyl-CoAs. Does anybody remember how many carbons an acetyl-CoA has?
3?
Two.
2 or 3?
2. Yeah, so pyruvate has 3.
Okay.
And then it gets acted on and it breaks it down a little bit more and you get 2. You actually get a release of a carbon dioxide in that first step going from glycolysis into the Krebs cycle. And so it has 2 carbons. Acetyl-CoA has 2 carbons on it. And so if we stop and we think about that, let's say if we have a fatty acid chain that has 18 carbons, how many acetyl-CoA are we going to have? Are we going to end up with?
9.
9, exactly. The tricky question there, and we'll get this again.
The number of steps would be 8.
Exactly.
Okay.
Because we have, let's say each of these represents an acetyl-CoA. We have an acetyl-CoA, an acetyl-CoA, an acetyl-CoA, like this. It's better to just draw it as 2 because otherwise it gets confusing. 1, 2, 1, 2, 1, 2. Where the bonds that we need to break are going to be here and then here. Because as soon as we break off the second one, we're left with 2 and we get 1. So we have 2 steps, but we get 3 acetyl-CoA. That's always a test question. Always, always, always. And you'll usually get 2 or 3 or 4 of them just like that. Of like, hey, how many steps is this? Or how many acetyl-CoA do you get? That kind of stuff.
Nice.
He'll usually throw in a lot of extra words. So just read the question. Sometimes it gets, because of a lot of the words, it seems a lot more difficult than it is. Okay, talking next step, so we have the fatty acids and they need to be in the matrix, so they have to cross the mitochondrial membrane and get into the mitochondria. So the mitochondrial membrane is impermeable, so it's not able to doesn't allow fatty acyl-CoA to pass through. Therefore, fatty acyl-CoA, big thing to notice here, acyl-CoA versus acetyl-CoA, they're going to be slightly different, okay?
Oh.
Just pay attention to that. A lot of test questions, that'll get you. It'll be like acetyl versus acyl.
I was about to say, can we go over that difference really quickly?
Yeah. So it's literally so it's kind of the this is like one of the lovely squares, a rectangle, but not all rectangles are squares kind of thing. So acetyl-CoA is a type of acyl-CoA, but fatty acyl-CoA is not acetyl-CoA. Okay. Acetyl-CoA is the very specific one. It's been acetylated in order to form the coenzyme A. So the actual, that one little step is what makes it acetyl-CoA. So if we think kind of more individualized is acetyl-CoA. More big picture is acyl-CoA.
Oh, okay.
Which kind of includes more than just the specific acetyl-CoA. So a lot of times you'll have answer choices where, let's see, when I find a good question, I'm going to write myself a note that when I find a good question about that, because he'll have different answer choices. Like he'll have fatty acyl-CoA and then he'll have acetyl-CoA. And like that's when you need to look at it and be like, okay, what are we talking about? Kind of where are we right now? Like, are we going from pyruvate through into the Krebs cycle, or are we doing more beta oxidation fatty acyl-CoA? Okay, like, are we more specific or are we more general?
And as I'm
Does that
looking
help?
at it's helping me. And I did find like a, the schematic that represents both acyl and acetyl. And what I notice is, at least from what I was able to see, the acetyl-CoA has a methyl group at the end. You got the CH3. That's the same one. And then the acyl, it's just an R group. So whether that's the fatty acid, like you were saying, or it could be other things. So it's whether it's an R group or a methyl.
Yeah. So just there can be a little bit of a difference right in here. So let's actually look up to see if we can have an example of fatty acyl-CoA.
Cool.
Give us
Yeah, I wouldn't have even. There's
But
like
yeah.
a part of me that just kept like superimposing those two as each other, and so I'm glad
Yeah.
you took a minute to just
So
talk about it.
try and keep them separate. Just read the questions carefully.
Yeah, thank you.
Because that's literally like a two letter difference between the two. So be careful, be conscious of that. And like I said, most of the time he's going to have it written as fatty acyl-CoA, so that helps a little bit as well. But sometimes he'll have an answer choice that's like fatty acetyl-CoA, and that's not a thing. So it's immediate. That's an immediate kind of cross that out. Fatty acetyl-CoA is not a thing, so we're not gonna focus on that.
Okay.
Okay. So the mitochondrial membrane doesn't let the fatty acyl-CoA through, so it must be activated by being bound to another protein called carnitine in order to cross the mitochondrial membrane. Remember, it has the two. This is just a nice little two different layers. The inner mitochondrial membrane has those folds known as cristae. Last trimester, did he ever have a question about cristae?
I can't remember.
I want to say yes.
I want to say no.
Okay. Some people, and like sometimes people get slightly different versions of an exam, so he might have. I always remember him, for some reason, I always remember him giving questions about cristae. So if you don't remember questions in Biochem 1 about cristae, watch out for them in Biochem 2. Just understand that that the cristae is part of the inner mitochondrial membrane. Don't forget about all the stuff that we talked about in Biochem 1, how you have the outer membrane, the inner membrane, the matrix, the intramembranous space, all that kind of stuff. Don't forget that we talked all about that. Okay. And then the inner membrane contains all these folds, and those folds are the cristae. So So before they're oxidized, the fatty acids must be transported across the mitochondrial membrane, and they must be activated by the addition of the CoASH or the coenzyme A before they can be metabolized. So this is now our actual form, and kind of we started to talk about it up here. That we're going to look at the actual making of acyl-CoA. So any questions up to this point? Feel like this is good. This is helping, kind of like seeing all the information, breaking it down a little bit.
Yeah.
Okay. So the fatty acids are activated by enzymes present in the outer mitochondrial membrane, and they produce acyl-CoA. This reaction is catalyzed by acyl-CoA synthetase. This enzyme is also called thiokinase, since they are making high-energy thioester linkages. That right there pops off a light in my brain saying that is a test question. The acyl-CoA synthetase is going to make what type of linkages? Thioester linkages. Or a question like thioester linkages are formed by which of the following enzymes? The acyl-CoA synthetase, aka thiokinase. I'd write down both of those, make sure you have those. Those are the same thing and what they do. So thioester bond is formed between a fatty acid carboxyl group, and the thiol group of coenzyme A, creating that fatty acyl-CoA. This is the only step that requires ATP as an energy source. Again, that's going to be a test question. Because if you need ATP, anything that requires ATP is going to be a rate limiting step, which means he likes to ask questions about it. So the major route of oxidative catabolism of fatty acids, we talk about beta oxidation, which occurs at the beta carbon atom of fatty acids. And the site of the beta oxidation of fatty acids is the mitochondrial matrix. Again, that right there. Promise it's some type of test question. It might be a little bit more specific, like it might list a step of beta oxidation. And that specific step is going to say, like, where does this take place? And it's going to have it within the mitochondrial matrix. Next question, transportation of fatty acids across the mitochondrial membrane, how it actually gets across. So the fatty acid transport in the mitochondria, we have small fatty acids less than or equal to 12 carbons are able to go across or go freely across the mitochondrial membranes. So the small fatty acids can go across. Anything larger need to go through the acylcarnitine or the aka the carnitine transporter protein. Yeah, that's going to be the main question there. Just understand that carnitine, the acylcarnitine or carnitine transporter protein, is what brings our larger fatty acids greater than or equal to 14 carbons. I remember this being a test question. This part here, kind of differentiating whether it would give you like the name or an example of a fatty acid, and you had to remember either like how many carbons it had, or it gave you the number of carbons, and then it would determine how does this pass through these membranes. And the answer choices that it gives you is either, you know, one of the different types of shuttles that we learned previously in Biochem 1, or like it diffuses freely, or the carnitine transporter protein. So remember those specific numbers. That small ones less than or equal to 12 can go right through. Greater than or more than 14 is going to be require the carnitine transporter protein. Another thing, all of our fatty acids are going to be in even numbers. They're going to be grouped compared to 2, because when we break them apart, they need to be in groups of 2, not any groups of 3. So if you see any answer choice that has an odd number of carbons, immediately cross it out, because it's not going to be one of those. The transport of the activated fatty acyl-CoA into mitochondrial matrix, if the role of the carnitine. Occasionally, he'll use the full name. I remember him at one point, either was a quiz question or test question, he used the full name of carnitine, so you needed to remember the full name of carnitine, or at least recognize it. Like if you can recognize beta hydroxy beta hydroxy gamma butyrate, remember those, like beta hydroxy butyrate. Look for those key words to hopefully be able to pick out the answer. The fatty acyl-CoA is then transported into the mitochondrial matrix by the help of carnitine. So carnitine through here, we have the carnitine acyltransferase 1. Gets it all the way through into the intramembrane space and gets it into another transport protein into the matrix. There's the 2 different proteins, the carnitine acyltransferase 1 on the outer part, and then the carnitine acyltransferase 2 on the inner mitochondrial membrane. I don't remember there being a ton of questions on this, but again, when I figure out the whole question debacle, we'll see more of those. I think what I'm going to try and do is I'm going to try and record a video later this week or next week, going over some of the first handful of questions, just so that way we can kind of see those, and so people can watch them before the test next week, because it's just because we got a little bit of a late start doing biochem 2. Clinical correlation. A lot of times these become test questions. Keep an eye on this. More than likely, it'll have something about a malonyl CoA be inhibiting the carnitine acyltransferase 1. Okay. Questions or any concerns up to this point? Is doing okay?
Yeah, just the my biggest question is, would you say just to memorize all the steps on the, like all the enzymes and the products for the exam, and just also for boards in general?
Yeah, unfortunately, that's kind of the way that it goes. If you're gonna memorize, best things to memorize, anything that's a rate limiting step or this like specific enzymes. A lot of this stuff doesn't, like the carnitine stuff doesn't typically end up on boards, or at least from what I have heard about boards and the boards that I've taken and stuff, I don't remember seeing anything about like this stuff super specifically. A lot of stuff in like biochem 1 is on boards, and then like biochem 2, it's a little bit more kind of hand-picked kind of stuff that you'll see on boards. So it's not a ton, but for test purposes, yeah, you'll want to remember the enzymes. If it gives like a specific location where they are, remember that, and then kind of in general what they do, names of stuff, where they are, what they do, like their reactants and products.
Okay.
Which it's a lot of memorization, but if you can find ways, to kind of break it down a little bit, like this picture looks really busy, but really all it's saying is, all you have to remember is that the carnitine acyltransferase 1, it's first, so it's on the outside, and the second one is on the inside, and so you get through the fatty acid through into the matrix. So
yeah, then Ninja Nerd goes over it pretty well.
Love Ninja Nerd. It's Ninja Nerd is great. Only issues with doing with like a specific course is that Ninja Nerd isn't the professor, but
Yeah.
Ninja Nerd is fantastic to learn a lot of these concepts and going through all these different steps. He does a really, really good job. Also for Phys 1, when you guys get to EKG stuff, I'd watch Ninja Nerd's. It's like an hour or something long, but he goes over like EKG stuff, and it's really, really good.
Okay. And then my other question is, anything that is like bolded, what do you say you pay special attention to that on the text? Like right
Typically,
there, like
yeah. Yeah, because Dr. Sarkar, he wrote this book, so anything that's bolded specifically tends to be, he likes to ask test questions on the bolded stuff.
Okay,
So,
gotcha.
So yeah.
All right. Well, thank you, Drake.
Yeah. Are you headed out?
Yeah, I'm heading out.
All right. Have a good night.
You too.
Yeah, I don't have any questions, so just gonna try and soak it all in.
Keep absorbing. Yeah. We'll go through the steps of beta oxidation. We're gonna take our time through them. Again, this is just gonna be, unfortunately, memorize the step by step, kind of what goes on, remembering the names of stuff. And these do kind of get confusing, but I'll try and point out the most important stuff, like palmitoyl-CoA. That's a big one, that we already mentioned previously. But let's kind of go step by step. So beta oxidation. So overall, big idea, beta oxidation, we're breaking down the chain, the fatty acid chain, into chunks of 2 to turn them into the acetyl-CoA. So the first step is beta oxidation, and it's called beta oxidation because it happens at the beta carbon, the second carbon of the fatty acid. So we have beta oxidation, then we have a hydration, another oxidation, and then a thiolysis. Those steps, oxidation, hydration, oxidation, thiolysis, just knowing the steps is going to be important for the test. So I do oxidation, hydration, oxidation, T. OHOT. It's going to be beta oxidation. So remember that, because that typically pops up as well. And also if you're trying to look at either like the order of the steps or trying to remember kind of what enzyme does what step, if you remember that the dehydrogenase enzymes do oxidation, hydration, and a thiolase, or a transferase or a thiolase, very similar. So we can remember the order of them. That'll help us remember the enzymes as well, so that we don't have to memorize each of the enzymes and their super long, boring names. So this different stages of beta oxidation. So the oxidative conversion, so converting that 2-carbon unit into acetyl-CoA from the carboxyl end of the fatty acyl chain with simultaneous production of NADH. So we're talking here, I think this actually might be typo. Okay, let's keep going. It might be a typo. I think it might supposed to be FADH, but I'm not 100% positive on that. Oh, no, I think this is just talking about in general, so no, it's fine with NADH. Stages. The second stage, oxidation of acetyl-CoA into CO2 via the citric acid cycle with generation of NADH and FADH2. So this is looking back a little bit more big picture. So this first part here, step 1, this is just the step that's referring to this, to the actual beta oxidation part, where we do have beta oxidation happen. We get acetyl-CoA, and then we remember from Biochem 1, acetyl-CoA goes into the Krebs cycle. So stage 2, that's our Krebs cycle. We get NADH and FADH2 out of the Krebs cycle. And then from there, it goes to the electron transport chain, where we get the making of ATP from the two of them through the electron transport chain. And then if we consider our fatty acid as the palmitoyl-CoA, so we're just looking at this example right here. It's a 16 carbon fatty acid. Then there would be a total of 7 cycles to remove a total of 8 molecules of acetyl-CoA. Remember, so we take the number of carbons, divide it by 2, and that gives us the number of acetyl-CoA. And then if we take the number of carbons, divide by 2, minus 1, in this case 7, that gives us the number of cycles, or the number of OHOT steps of these steps right here, the dehydrogenase, hydratase, dehydrogenase, thiolase steps had to occur in order to get all 8 molecules of the acetyl-CoA off. He likes that as test question. Beta oxidation of saturated fatty acids, sequential steps. So we have 4 enzyme catalyzed reaction steps. Removes 1 acetyl-CoA at a time from the COOH. Remember, that is also known as the carboxyl type here. Carboxyl end of the fatty acyl chain. So 4 enzymes of each pass remove 1 acetyl-CoA at a time. This is just repeating these steps up there in words. The dehydrogenase, which is an oxidation, hydratase, which is a hydration, dehydrogenase, oxidation, and then thiolase, rearranging. And here's just more in words, the picture up above. I personally, if you can draw the picture just with the words, it's like I would start to do PCOA, arrow, oxidation, T delta 2 ECOA. Break these down, abbreviate a little bit. Most of the time with his test questions, you can get it just from knowing that rough thing, like the rough translation, not like the every single word, memorizing every single word. So I prefer to draw it out than to read all of this. So, and all of this is just a repeat of what we talked about. Another thing he might ask is more kind of simple math stuff, whereas how much ATP gets made. So all the acetyl-CoA generated through the oxidative, all the acetyl-CoA that's generated from breaking down palmitoyl-CoA, remember that's 16 carbons, enters the citric acid cycle. Remember, each turn of the citric acid cycle, it was 3 NADH plus 1 FADH2 Remember, each of the NADHs gives us 2.5 ATP. Each FADH gives us 1.5 ATP. So by going through all of the stuff, electron transport chain, it generates 129 ATP molecules. We won't have you do big math typically, but his big idea is he'll ask something along the lines of showing that fatty acids produce way more ATP than glucose ever does. Overall equation. You don't need to memorize all of that. You know, just remember the steps. Fatty acid catabolism. This also includes, because within here we develop an NADH and an FADH2 from each of the turns. Those also go off to make energy. So it's more than that. So we actually technically get 4 NADH and 2 FADH2 with each of them. Because we also make NADH and FADH2 actually creating the acetyl-CoA. The acetyl-CoA enters the citric acid cycle, further oxidizing CO2, makes GTP, NADH, FADH2, all that stuff. And here's just the math that it does out for you. This isn't super important. I don't remember him. This might correct something that I said earlier where it needs to be even number. Most of what I remember ever dealing with in this course was even number. I don't remember him ever giving us odd number chain stuff. But if he does ask a question about it, so I'm just going to step back what I said previously. If it's talking about breaking it up into acetyl-CoA, and there's an even number, it's always just going to be acetyl-CoA. But if for whatever reason he gives you an odd number, which I don't remember him giving a lot of odd numbers, you also end up with a propional CoA at the last cleavage because we have an extra carbon. We'll have 3 carbons instead of 2 remaining. That's like the only specific question to ask about that. Again, he doesn't really give you any odd ones really to know. So, you doing okay, Aaliyah?
Yep, as okay as I can.
Yeah. Okay, so talking about different types of fatty acids, unsaturated versus saturated. And there's a couple different terms. You know, we have, when we talk about fats, we have monounsaturated, polyunsaturated, and then we have saturated fats. What we're referring to when we talk about saturated is the number of hydrogens that are bound onto everything. Okay, so like right here, this double bond right here, because there's one double bond. Mono is 1 unsaturated. What that means is that there's not hydrogens bound to those carbons. So earlier when we drew the carbon chain like this, carbon, 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 and then we had 2 hydrogens on each of these. And unsaturated looks like this. We remove these hydrogens and instead form a double bond between the carbons. Which just, if we have unsaturated, let's say we have, like, this is a monounsaturated, but I have polyunsaturated, we'd have to do this extra times. And there's a rule. There's the rule. Let me look it up. I'm sure we'll get to it. There's a certain rule in humans where they can't create double bonds past a certain level. So we'll get to it when we get to it. So the extra step is that we need an isomerase to rearrange this and add on hydrogens in order to perform the beta oxidation. So just remember this extra little step. We need an isomerase because we need to rearrange this and add in those extra hydrogen so that it's fully saturated so that we can then break it down. Enoyl-CoA isomerase is for monounsaturated. NADPH dependent 2,4-dienyl-CoA reductase. Super long name. The names look really, really intimidating. Best way to do this material is to create a Quizlet and literally just have this name, and then on the flip side of the flashcard say it's enzyme for polyunsaturated fats, because that's going to be the best way to just honestly raw recognition and memorization of these guys. We have a couple different kinds of oxidation. So most of the fats we talk about beta oxidation. So in general, most of what our body does is beta oxidation, but we do have some extra ones. There's alpha oxidation of fatty acids, which happens in the brain, and that's removing one carbon at a time. There's omega oxidation, occurs in microsomes involving cytochrome P450. Don't worry too much about all of this stuff. The cytochrome P450, that will come back later on. I don't remember if we talked about that in Biochem 1 or if it's in eicosanoid stuff in Biochem 2, but this will come back later. If he asks it, I would just, the only thing I do, alpha, remember brain, omega, cytochrome P450. That's really all you need to remember. Peroxismal fatty acid oxidation differs from the mitochondrial oxidation, where we have very long chain fatty acids from a high fat diet are oxidized to peroxisomes. We don't really go in deep into what peroxisomes stuff are, so that's all you really need to know. High fat diet, you get peroxisomes. Left here. Ketones. We'll go ahead and cover this real quick just so we finish the chapter. So the synthesis of ketone bodies, remember we talked at the very beginning, the ketone bodies when we have like diabetes or we're in a starvation state and we have no more glucose to break down and to use. We make ketone bodies to substitute to keep ourselves alive. And that occurs in the liver mitochondria when fatty acid concentrations are high in the blood, either during fasting, starvation, untreated diabetes, or with a really high fat diet. 2 fates of acetyl-CoA produced during beta oxidation of fatty acids. The acetyl-CoA enters the citric acid cycle, or you can get ketogenesis, which is an alternative pathway. So we have acetone, acetoacetate, and then D-beta hydroxybutyrate. These are our 3 ketone bodies. Unfortunately, this is just another step-by-step that you need to write out, memorize, learn it well enough. We take 2 acetyl-CoA. The thiolase gives us acetoacetyl-CoA, so it combines them. You get HMG-CoA synthase to create HMG-CoA. Then HMG-CoA lyase breaks it back down into acetoacetate, which then gives us acetone and our D-beta hydroxybutyrate, which are our ketone bodies. Again, unfortunately, This is just more memorization of step by step. Very unfortunately, but that's what it is.
So is it more beneficial to just know the names and then have a general idea of the structure?
You honestly don't need to know the structure. Like he's not going to ask you, he's not going to show you pictures of structure and like, what is this? More than anything, it's the names and the enzymes. Like knowing that acetyl-CoA gets acted on by a thiolase, 2-acetyl-CoA gets acted on by thiolase to make acetoacetyl-CoA. And then acetoacetyl-CoA gets acted on by HMG-CoA synthase to make HMG synthase. And then that gets cut to make acetoacetate. And then you have acetoacetate decarboxylase or hydroxybutyrate dehydrogenase to go to either of these. So it's easiest to remember. So you can honestly take out like all of this, all of like the actual drawings, simplify it a little bit more for yourself. And when you do that, when you take out all the drawings, it looks a little less intimidating, I think. First thing you want to remember, most important, is going to be kind of the steps and the enzymes. So this to this, using this enzyme. This to this, this to this, using this enzyme. The next extra thing, if you have the time to add it in, would then be to add in like the extra stuff off to the side. If you have the time and the brain power, most of your questions are going to come specifically from what I was saying, the reactant to product and the enzyme. That's majority of the questions that come from there. So it's, and with, you know, how busy Try2 is, just kind of prioritizing the information is really, really important. So that way you can get the most benefit out of it. And then this whole last portion of all of this, stuff that we've already talked about, just when ketone bodies are produced in diabetes with starvation. And then if you have too many ketones, you get ketoacidosis or ketosis. So a lot of extra words on this page just to say what I just said. And that's all of chapter 1. So we got through all of chapter 1 in about an hour and a half, a little over an hour and a half, hour 40. That's kind of, so from here, you think exam 1 is chapters 1 through 4. So right now, start thinking, I know you have a lot of exams coming up. Start budgeting your time to understand, okay, before biochem, you need to dedicate, give or take, hour and a half, or maybe a little bit less. You can also watch the YouTube videos that I already have out on these chapters. Go through each of the chapters. And then I'm going to try and make the videos for the Linus Learning questions, or maybe next week tutoring session, we'll just, once I figure out the order of stuff, we'll just crush through a bunch of the Linus Learning questions to prep for tests. So, and hopefully, I think you said are all of your exams Monday, Tuesday, Wednesday, or do you have anything on Thursday?
At least for Try2, it's micro and physiology on Wednesday, and then gross anatomy on Thursday, I believe. Let me look at my calendar.
Okay. I think, are you blended or on campus?
Blended.
Okay. I think they're a little bit different. I think on campus has micro and phys on Tuesday, and then gross on
Yes.
Wednesday, I think.
So I might be wrong, but I have it down as physiology and gross anatomy on Wednesday the 29th, and then micro on Friday the 31st.
On Friday, okay. So we'll try next week. We'll try and go through, and I'm gonna come back through and spend some time, probably reread this chunk of the book as well, and we'll hit a lot of the questions and do some of the prep work for the upcoming Biochem 2 exam, which happens next time. So the week after. All right, Aaliyah, do you have any other questions or anything else I can help you with?
Not that I can think of.
Okay. How are you doing with school and everything going on?
I mean, it's a lot.